Welcome to this explainer on the complex embryological development of the pancreas and midgut. Now, if you're a second-year medical student, you already know that embryology can sometimes feel like trying to visualize this wildly intricate, multidimensional puzzle in your head. It's tough. But today, my goal is to give you a precise, textbook-level understanding of this morphologic journey. We're going to unpack the exact developmental stages, the mechanisms, the derivatives, and of course, the clinical correlations you absolutely need to master this material. So, grab a coffee, and let's dive right into this.
All right, here's our roadmap for today. We'll be using a system-based progression. We're starting with the localized development and fusion of the pancreas, mapping out its ductal systems and looking at the clinical abnormalities. Then, we'll expand our scope distally to the midgut, tracking its massive elongation, its dramatic physiological herniation, its rotation, and finally, its retraction back into the abdominal cavity.
Section 1. Pancreas Development and Fusion. Let's get right into the foregut endoderm and check out how these initial buds fuse together.
Okay, let's look at the initial outgrowths. You've got the ventral pancreatic bud, which actually shares a ductal connection with the developing biliary system, and the dorsal pancreatic bud. Both of these are direct outgrowths of the foregut endoderm. Initially, they are sitting on completely opposite sides of the developing duodenum. But, embryology is all about movement. Because the duodenum itself undergoes a 90-degree clockwise rotation, the ventral bud is mechanically forced to swing dorsally. It physically sweeps around to the back and tucks right underneath the dorsal bud. Then they fuse together to form your definitive single adult pancreas.
Now, understanding what these two separate buds ultimately become is absolutely critical. It is a classic, highly tested board exam concept that the ventral bud exclusively forms the uncinate process and just the lower portion of the head of the pancreas. That's literally it. The dorsal bud, on the other hand, is the heavy lifter here. It's responsible for the entire remaining gland. We're talking the rest of the head, the entire body, and the tail of the pancreas.
Moving right along to Section 2, Pancreatic Ducts and Papillae. We know how the tissues fuse, so let's look at the internal plumbing.
Initially, you have two entirely separate ductal systems for these buds. But when the buds fuse, their internal ducts have to anastomose, right? The main pancreatic duct, which you probably know as the ductus versum, is actually a composite structure. It's built by fusing the distal part of the dorsal pancreatic duct together with the entire ventral pancreatic duct. But wait, what happens to the proximal part of that dorsal duct? Well, it usually either completely obliterates or it persists as a smaller secondary channel that we call the accessory pancreatic duct, or the duct of Santorini.
So, how does this all connect to the gut tube? The main pancreatic duct joins forces with the bile duct, and together they empty into the duodenum at the major papilla. Now, if that accessory duct of Santorini is present, it gets its own little exit slightly superiorly at the minor papilla, and here's a massive clinical pro for you. In about 10% of cases, this fusion of the ductal systems completely fails. That means the original separated double system persists right into adulthood.
That perfectly segues into Section 3, Clinical Pancreatic Abnormalities. What happens to the patient when this morphological dance goes wrong?
The absolute crucial condition to understand here is the annular pancreas. Remember how that ventral bud is supposed to migrate completely to the right and dorsal side of the duodenum? Well, in an annular pancreas, the ventral bud tissue essentially gets confused. It splits or migrates in both directions and ends up fusing with the dorsal bud both dorsally and ventrally. The result? It creates a literal ring, an annulus of pancreatic tissue that tightly wraps around the duodenum like a belt. This is a severe morphological error, and it leads to massive duodenal obstruction in the newborn.
Here's another fascinating anomaly, ectopic or accessory pancreatic tissue. You can actually find random patches of pancreatic tissue anywhere along the digestive tract, literally, from the distal esophagus all the way down to the primary intestinal loop. Clinically, though, you'll most frequently see it popping up in the mucosa of the stomach or in a Meckel's diverticulum. And what's really wild is that this accessory tissue isn't just a dummy mass, it is fully functional. It can show all the histological characteristics and secrete the same exocrine and endocrine products as the main pancreas itself.
All right, transitioning to Section 4, midgut, primary intestinal loop. We are moving distally down the gut tube now into a phase defined by explosive, rapid growth.
I want you to picture the abdominal cavity of the embryo at this exact stage. The gut tube and its mesentery are elongating so rapidly they form this giant primary intestinal loop.
But simultaneously, the liver is undergoing massive expansion, so we have a serious real estate crisis on our hands. How on earth does the developing embryo accommodate all this growth in such a confined, tiny space? Well, this spatial constraint forces a brilliant physiological workaround that is absolutely crucial to midgut development.
The embryo's solution to this real estate problem is physiological gut herniation. That rapid elongation forms a massive U-shaped primary intestinal loop, and if you look at the very apex of this loop, it remains an open connection with the yolk sac through a very narrow, distinct tube, called the vitelline duct. You can think of this vitelline duct as the crucial anatomical pivot point for everything the midgut is about to do.
But before we watch this loop move, you must have this exact derivatives map locked down for your exams. The vitelline duct divides our U-shaped loop into a cephalic limb and a caudal limb. The cephalic limb, which is cranial, develops into the distal part of the duodenum, the entire jejunum, and part of the ileum. The caudal limb, on the other hand, forms the lower portion of the ileum, the cecum, the appendix, the ascending colon, and the proximal 2/3 of the transverse colon. You absolutely have to know these origins cold.
Section 5. Physiological herniation and rotation. Let's track the actual dynamic, physical journey of this elongating gut as it rotates out of and eventually back into the abdomen.
Timing is absolutely everything in embryology. Because of that rapid growth of the intestinal loop and the massive liver expansion we just talked about, the intestines literally run out of room inside the body. So during the 6th week of development, they are forced to physiologically herniate. They actually push out into the extraembryonic cavity right inside the umbilical cord. And they're going to hang out there, continuing to grow and develop until the 10th week when they finally start their journey back home.
Now, while this loop is pushed out into the umbilical cord, it isn't just resting. The small intestine continues to elongate rapidly, forming a bunch of highly coiled jejuno-ileal loops. At the exact same time, the entire midgut is undergoing a highly specific, multi-stage rotation around a central vascular axis. It's a beautifully choreographed sequence of herniation, coiling, rotation, and eventually retraction.
Let's break down the exact math of this rotation. The primary intestinal loop needs to rotate a grand total of 270 degrees counterclockwise. The physical axis it spins around? The superior mesenteric artery. Now, this doesn't happen all at once. It happens in 2 stages. First, there's a 90-degree counterclockwise turn that occurs while the gut is herniating out into the umbilical cord. Later, the remaining 180-degree turn happens during the return of the intestinal loops back into the abdominal cavity. Add them up, and you get your 270 degrees.
Which brings us to our final phase today, Section 6, Midgut Retraction and Fixation. We've hit the 10th week, and the midgut is ready to complete its journey and settle into its permanent anatomical home.
So what changes at week 10? Why is the gut suddenly able to fit back inside the body? Well, the abdominal cavity finally has the space to welcome these loops back because of 3 simultaneous events. First, the large mesonephric kidney significantly regresses. Second, the massive growth rate of the liver finally slows down. And third, the abdominal cavity itself has simply expanded as the embryo has grown.
This return process is incredibly orderly. The proximal portion of the jejunum is the very first part to re-enter the abdominal cavity, and it predictably settles right over on the left side. As the rest of the loops follow, they gradually pack in, settling more and more toward the right. Finally, the cecum and the colon return last, rotating to their definitive adult positions on the right side of the abdominal cavity, forming a neat border that beautifully frames all those coiled loops of the small intestine.
As we wrap up today's explainer, I want to leave you with a crucial clinical puzzle to chew on. Once these loops return to the abdomen, they have to fuse and fixate to the posterior body wall. But if the returning colon experiences incomplete attachment, what dangerous clinical vulnerabilities does that create for the patient? Understanding this flawless, stepwise embryological journey is the absolute key to recognizing the catastrophic effects of intestinal malrotation and volvulus when you're out there on the clinical wards. Keep studying these stages, make sure you master those derivatives, and I'll see you in the next explainer.
Hey everyone, and welcome back to this explainer. Today we're tackling the midgut and hindgut. I know you guys have been grinding through embryology, and it is a beast, but we're going to master the second half of this complex topic together. We'll break down these mechanisms step by step, so you're not just memorizing, You're actually going to understand the why for your board exams and the wards. So let's just dive right in. All right, here's our game plan for today's explainer. We've got 6 key areas. First, abdominal wall defects. Second, vitelline and rotation anomalies. Third, gut atresias and stenoses. Fourth, hindgut and cloaca formation. Fifth, anal canal embryology, and finally, we'll wrap it all up with a high-yield exam review. Let's get to it. Starting off with Section 1, abdominal wall defects. Okay, so what happens when that normal physiological herniation of the midgut just doesn't resolve? Well, that's when we see an omphalocele. The absolutely crucial thing to understand here is that the bowel loops basically fail to return to the body cavity. They just stay out there, herniating right through an enlarged umbilical ring. To put some numbers to it, this happens in about 2.5 out of every 10,000 births. And clinically, it's a really big deal. It comes with a high mortality rate, around 25%, mostly because it's so frequently tied to other really severe developmental malformations. Take a look at this image. It brilliantly illustrates the pathognomonic feature of an omphalocele. See how the herniated intestinal loops are physically covered by this protective amnion membrane? That translucent sac encasing the bowel right at the midline? That is your defining diagnostic feature. You absolutely have to commit that visual to memory. Now here's where it gets really testable. Board examiners absolutely love making you differentiate an omphalocele from our next defect, gastroschisis. With gastroschisis, the abdominal contents protrude directly into the amniotic cavity. The key difference? It happens lateral to the umbilicus, usually on the right side, and there is zero covering membrane. No peritoneum, no amnion, nothing. Because the bowel is just sitting out there exposed directly to the amniotic fluid, it can get damaged. But, interestingly enough, gastroschisis usually isn't linked to severe chromosomal abnormalities. So despite how it looks, the overall survival rate is actually excellent. Let's put them side by side to really cement this in your brain. On the left, you've got the central, membrane-covered omphalocele pushing right through the umbilical ring. And on the right, contrast that with the lateral, exposed, completely uncovered bowel of gastroschisis. That right-sided defect is actually thought to be caused by abnormal body wall closure around the connecting stalk. Take a mental snapshot of this comparison right now, guys. It is incredibly high yield. Moving right along to Section 2, vitelline and rotation anomalies. So the vitellin duct is supposed to completely obliterate, right? Well, when it doesn't, things get complicated, and it can have absolutely devastating vascular consequences for the fetus. For instance, you might see a vitellin cyst, also known as an enterocystoma. This is where a cyst literally remains attached to both the umbilicus and the wall of the ileum by vitellin ligaments. Or you could get a vitellin fistula. That's an open tract connecting the lumen of the ileum directly out to the umbilicus. And on top of that, we have to talk about overall gut rotation defects. If the gut doesn't rotate properly, it can cause volvulus. That's a twisting of the intestine that can severely compromise the vital blood supply, leading to massive tissue death and potentially fetal death. Blood supply is literally everything for the growing GI tract. All right, Section 3, gut atresias and stenoses. Let's shift our focus to the inside, the internal lumen of the gut tube. What happens when the normal microscopic hollowing out process just fails? Well, you get atresias and stenoses. Now, these can happen anywhere from the distal portion of the duodenum all the way down, but they happen most often in the duodenum itself, with the fewest occurring in the colon. Upper duodenal atresias are super heavily tested, and what causes them? A failure of recanalization. Recanalization is this amazing microscopic process where the gut tube, which actually starts out as a solid cord of epithelial cells, develops these little vacuoles that merge together to hollow it back out. This visual perfectly captures that morphological shift. Look at figure A on the left. That's the solid stage. The epithelial proliferation has completely blocked the lumen. Now contrast that with figure B on the right. See how it's actively hollowing out? Those little vacuoles are coalescing to rebuild that continuous internal pathway. If this process on the right stalls out or fails entirely, you're left with either an atresia, which is a total roadblock, or a stenosis, which is a severe narrowing of the tube. Let's head further south. Section 4, hindgut and cloaca formation. Continuing our journey caudally, we're mapping out the exact territory of the hindgut. Fed by the inferior mesenteric artery, the hindgut gives rise to a lot. The distal third of the transverse colon, down through the descending colon, the sigmoid, the rectum, and the upper 2/3 of the anal canal. But here is a super crucial detail you cannot forget. The endoderm of the hindgut also impressively forms the internal lining of the bladder and the urethra. It's a really cool crossover. Okay, so think about this. How does one single posterior tube manage to split into separate gastrointestinal and urogenital tracts? It's all about a very dynamic timeline. Initially, you have the primitive anorectal canal entering the back of the cloaca, and the allantoids entering the front. To separate them, a wedge of mesoderm, called the urorectal septum, starts to descend right between the allantois and the hindgut. This dramatic descent ends with the cloacal membrane rupturing right at the end of the 7th week. Finally, the very tip of that urorectal septum anchors permanently in place, and that anchored tip, that becomes the perineal body. If you trace this process visually, you can see the urorectal septum acting just like a wedge, driving relentlessly downward. It is physically splitting the cloaca in two, pushing the primitive urogenital sinus to the front and isolating the anorectal canal to the back. And as that separation advances and the cloacal membrane finally ruptures, look at what we're left with. We get a ventral opening for the urogenital sinus and a dorsal opening for the anus. Check out exactly where that urorectal septum anchors. It sits perfectly as the perineal body, right between these two newly separated, totally distinct functional systems. It's like an anatomical cornerstone. Moving on to Section 5, Anal Canal Embryology. All right, we've reached a massive board exam favorite, the pectinate line. This highly tested landmark represents the exact anatomical junction where endoderm from the hindgut beautifully meets ectoderm from the proctodeum. It is a strict, definitive dividing line. Everything changes here: the type of epithelium, the arterial supply, the venous drainage, and even the lymphatic drainage. It's literally a border between two different embryonic worlds. You absolutely want to take a mental screenshot of this atlas-style table. Distinguishing these two embryological halves is non-negotiable for exam day. The upper anal canal, the cranial part, comes from endoderm. It's lined with columnar epithelium and gets its blood from the superior rectal artery. Now flip to the lower anal canal, the caudal part. It comes from ectoderm around the proctodeum, is lined by stratified squamous epithelium, and is supplied by the inferior rectal arteries, which, by the way, branch off the internal pudendal arteries. You gotta know this inside and out. And finally, Section 6, High-Yield Exam Review. Let's consolidate everything we just talked about. Lower GI embryology is really just this delicate dance of macroscopic folding, like midgut herniation, and microscopic hollowing, like recanalization. When those steps fail, that's when we see severe structural anomalies like omphalocele, gastroschisis, and atresias. And it all culminates in that incredible tissue convergence down at the cloaca and the dual origin anal canal. Here are your 5 golden revision points to memorize for the boards. Number 1, omphalocele has an amnion covering, gastroschisis does not. Number 2, gastroschisis is lateral, usually on the right, and generally isn't linked to severe chromosomal defects. Number 3, upper duodenal atresia is born entirely from failed recanalization. Number 4, the cloacal membrane ruptures late in the 7th week. And number 5, the pectinate line perfectly separates endoderm and the superior rectal artery above from ectoderm and the inferior rectal artery below. To wrap things up, let's talk about applying this to clinical vignettes. Train your eyes to hunt for keywords. If you see amnion, think omphalocele. If you see exposed bowel, think gastroschisis. Always, always link recanalization failure directly to atresias. And structurally, when you're looking at the anal canal, just remember, above the pectinate line is superior for blood supply, and below is inferior. So before we end this explainer, ask yourself, can you confidently trace the timeline of a single embryonic cell from a solid gut tube all the way down to its final adult home in the anal canal? Master that journey, and you've absolutely mastered GI embryology. Keep studying hard, you guys are doing great, and I'll see you in the next explainer. Welcome to this explainer. Today we're tackling a massive, highly testable board exam topic for second-year medical students. We're going to chronologically trace the complex embryological development of the human kidney systems. By the end of our time together, you'll have a rock-solid understanding of how we go from a basic layer of embryonic tissue all the way to a fully formed, highly complex urinary system. Okay, let's dive right in. To build a clear mental map, we're going to structure our review sequentially today. We'll start with the origins in the intermediate mesoderm, and then progress through the pronephros, mesonephros, metanephros, and finally the uroderic bud subdivisions. All right, let's jump right into Section 1. Origins, the intermediate mesoderm. Okay, so here's the fundamental rule you've got to commit to memory right off the bat. Both the urinary system and the genital system actually develop side by side, and they come from a shared common origin, a mesodermal ridge called the intermediate mesoderm. Why does this matter? Well, the fact that these two massive systems arise from the exact same tissue explains why clinically developmental anomalies in one system frequently show up with concurrent anomalies in the other. It's a package deal. Take a look at this transverse section of the early embryo. To orient yourself, First, find that central notochord. Got it? Okay, now move laterally, right past the paraxial mesoderm. You'll clearly see the intermediate mesoderm sitting right there, positioned just before the lateral plate splits into its somatopleuric and splanchnichloric layers. This exact anatomical spot, that's the starting point for our entire kidney story. Now, let's look at the timeline, because you definitely want this anchored in your memory. Human kidney development is basically a story of 3 overlapping systems, and they appear strictly in the 4th and 5th weeks of development. It goes like this. Early 4th week, the pronephros appears. Late 4th week, the mesonephros takes over and even functions briefly. And finally, in the 5th week, the metanephros shows up. Let's break these down one by one. Moving on to section 2, pronephros, the rudimentary kidney. If you look at the sagittal view here, notice how the pronephros is situated way up high. It's represented by just 7 to 10 solid cell groups located exclusively up in the cervical region. Now, checking out the cross-section, you'll see it actually goes as far as forming internal and external glomeruli, plus a nephric tubule. But, and here is the key fact for your exams, this first system is entirely rudimentary. It is completely non-functional in humans. And by the end of the 4th week, poof, all indications of this pronephric system have disappeared entirely. It basically paves the way and then just vanishes. Which brings us to section 3, mesonephros, the interim system. So, as that first system vanishes, our second system starts forming further down the embryonic axis, right in the thoracic and lumbar regions. This is the mesonephros, and it's significantly larger than its predecessor. It features these really distinct mesonephric excretory units, think of them as early nephrons, and it develops its very own collecting structure, which we call the mesonephric or Wolffian duct. In the human embryo, this actually acts as a brief interim kidney during that early fetal period. All right, listen up, because here is a classic, super high-yield board exam concept you absolutely cannot afford to miss. While the mesonephros itself only functions briefly as a kidney, its primary duct, that mesonephric or Wolffian duct we just talked about, has a strictly sexually dimorphic fate. What does that mean? Well, in males, a few of the caudal tubules and the mesonephric duct actually persist, and they go on to participate heavily in forming the male genital system. But in the female embryo, these structures disappear completely. Remember earlier when we said the intermediate mesoderm gives rise to both the urinary and genital systems? This right here is a prime example of that shared developmental destiny. Next up, section 4, metanephros, the permanent kidney. Now we're in the 5th week, and I want you to notice this crucial interaction happening right down near the cloaca and hindgut. See that tube-like outgrowth? That's the ureteric bud, and it's penetrating a dense mass of tissue called the metanephric blastema, which comes from unsegmented mesoderm. The definitive excretory units, the actual nephrons of your permanent kidney, develop directly from this metanephric blastema, but only because of its close association with that invading ureteric bud. Literally, if this interaction fails, the permanent kidney just does not form. It's a total deal breaker. And that leads us perfectly into section 5, ureteric bud subdivisions. You're going to want to watch this exact sequence of events because it's highly testable. The ureteric bud isn't just a static tube, right? Step 1, the bud first dilates to form the primitive renal pelvis. Step 2, this pelvis splits into distinct cranial and caudal portions, and these will become your future major calyces. Then step 3 is kind of fascinating. As the branching continues, the tubules of the second order actually enlarge and absorb the tubules of the 3rd and 4th generations. This specific process of absorption is exactly what forms the minor calyces of the renal pelvis. But wait, the process doesn't stop there. This branching mechanism is absolutely relentless. The ureteric buds just keep subdividing, driving the formation of 12 or more generations of tubules. Plus, even more tubules continue to form and grow out at the extreme periphery of the kidney, all the way until the end of the 5th month of gestation. It's just a massive ongoing period of cellular proliferation and architectural organization. So to firmly synthesize this for your exams, you need to permanently link the ureteric bud to these specific anatomical derivatives. If a board question asks you about the origin of the collecting system, you are looking for the ureteric bud, for sure. It specifically gives rise to the ureter itself, the renal pelvis, the major calyces, the minor calyces, and the vast network of collecting tubules. Drill that list into your head. And to truly appreciate the sheer scale of this embryological feat, let's look at the numbers. Because of that relentless generation after generation subdivision we just went through, a single tiny embryonic ureteric bud ultimately branches enough times to produce between 1 and 3 million individual collecting tubules per kidney. Honestly, it's just a masterpiece of biological engineering unfolding in a matter of weeks. As we wrap up this explainer on the origins and systemic development of the human kidney, I want you to think ahead to your next study block. We've successfully built this vast collecting system from the ureteric bud, right? But how exactly does that unsegmented metanephric blastema physically transform to cap these millions of tubules? How does it epithelialize to create a fully functional human nephron? What molecular crosstalk dictates that final critical step? Keep those questions in mind as you continue your embryology studies. Thanks so much for joining me on this deep dive, and keep up the great work. Welcome back to this explainer. Today, we're diving right into the morphological progression and the absolute highest yield clinical pathologies of the renal system. If you're a second-year medical student prepping for your embryology board exams, you are exactly where you need to be. We're going to master these complex developmental stages step by step together. Here's a quick look at our roadmap for today. First, formation of the nephron. Second, origins and developmental timeline. Third, renal agenesis and Potter sequence. Fourth, cysts, tumors, and ectopia. And finally, a high-yield exam review to tie it all together. All right, let's jump in. Section 1, formation of the nephron. Take a look at this progression. It's kind of like a giant puzzle coming together. If you check out the early stages on the top left, notice how each newly formed collecting tubule, which is in yellow, is distinctly covered by that blue cluster of cells. That right there is the metanephric tissue cap. This exact physical relationship between the tubule and the tissue cap is literally the foundational starting point for building a working kidney. So, let's break this transformation down step by step. First off, the cells of that metanephric tissue cap organize themselves to form these small, hollow structures we call renal vesicles. Next, those vesicles elongate into very distinct S-shaped tubules. And that shape, it's absolutely critical, because capillaries are going to grow directly into the pocket at one end of that S. Those capillaries will then differentiate into the glomeruli. Finally, the proximal end of each nephron forms Bowman's capsule, which gets deeply indented by that newly formed glomerulus. And boom! Together, this whole tubular system and its glomerulus form the nephron, our definitive excretory unit. Moving right along to Section 2, Origins and Developmental Timeline. Okay, this is absolutely crucial. You literally have to commit this to memory. The kidney develops from two completely distinct embryonic sources. The metanephric mesoderm is entirely responsible for providing the excretory units, so the nephrons we just talked about. On the flip side, the ureteric bud gives rise strictly to the collecting system. Do not mix these up. If an exam question asks you about the origin of a collecting duct versus, say, a proximal convoluted tubule, you have to rely on this strict division. Now, let's anchor our developmental timeline with 3 major milestones. First, early in gestation. Specifically by the 10th week, the glomerular capillaries differentiate, and believe it or not, urine production actually begins. Fast forward to birth. Nephron formation stops here, leaving the newborn with roughly 1 million nephrons in each kidney. At this stage, the kidneys have a really distinct, bumpy, lobulated appearance. But during infancy, that lobulation naturally disappears. Why? Well, actually, scratch that. It's not because the body is making new nephrons. It isn't. It's strictly because of the further growth and hypertrophy of the existing nephrons. Section 3, renal agenesis and Potter sequence. So what happens when these developmental steps fail? First up is renal agenesis, which is when the kidney just fails to form. Looking at the numbers, unilateral agenesis happens in about 1 in 1,000 births. Clinically, this can be totally asymptomatic because that single functioning kidney compensates beautifully. It just picks up the slack. However, bilateral agenesis is much rarer, about 1 in 10,000 births. And without any functioning kidneys at all, this results in catastrophic, fatal renal failure for the newborn. And that total lack of renal function in utero brings us to the Potter sequence. Now, you should conceptualize the Potter sequence not as just some random collection of symptoms, but as a direct, mechanical domino effect. It's a cascade of devastating fetal anomalies that stem entirely from that initial bilateral renal agenesis. Here is the exact cause and effect cascade for your exams. Because there are no kidneys, the fetus experiences anuria, a complete failure to produce urine. And since fetal urine is a massive component of amniotic fluid, anuria directly leads to oligohydramnios, which is a severely decreased volume of amniotic fluid. Without that fluid volume to physically expand the uterine cavity and let the fetal chest practice breathing movements, the fetus develops hypoplastic, severely underdeveloped lungs. So just remember the flow. Agenesis causes anuria causes oligohydramnios, and oligohydramnios causes hypoplastic lungs. All right, Section 4, Cysts, Tumors, and Ectopia. Visually contrast these gross external and cross-sectional views of autosomal recessive polycystic kidney disease, or ARPKD. This is a severe progressive inherited disorder happening in roughly 1 in 5,000 births. Notice how the normal kidney tissue is just completely replaced by numerous fluid-filled cysts. The incredibly high yield fact here, in ARPKD, these cysts form strictly from the collecting ducts. Just briefly, we absolutely have to mention Wilms tumor. While it's often seen in childhood, this mutational issue can actually begin developing in the fetus. It's a major pediatric kidney cancer that is highly associated with other developmental abnormalities and genetic syndromes. It's a real game changer for integrated clinical scenarios on the boards for sure. Finally, let's talk drainage tracts. In rare structural cases, one ureter might correctly open right into the bladder, while the other is ectopic. Because of developmental errors with the ureteric bud and the urogenital sinus, this ectopic ureter completely bypasses the bladder sphincter and drains abnormally. In females, these aberrant drainage sites typically include the vagina, the urethra, or the vestibule. And clinically, how does that show up? Often as continuous urinary incontinence. Almost done. Section 5, High-Yield Exam Review. Let's lock in our top 5 takeaways. 1. The metanephric mesoderm forms the excretory units. 2. The ureteric bud forms the collecting system. 3. Fetal urine production begins right at the 10th week. 4. The Potter sequence is a mechanical cascade going from anuria to oligohydramnios to hypoplastic lungs. And 5. The cyst in autosomal recessive polycystic kidney disease originates specifically in the collecting ducts. I'm going to leave you with this classic, highly tested trick question so you never get caught off guard. Remember, the kidney smooths out and loses its lobulation during infancy entirely due to the growth of existing nephrons, not from creating new ones past that 1 million mark. So why does understanding this hard cellular limit matter so much for adult renal pathology decades later? Think about that as you review your notes. Keep studying hard, and I'll catch you in the next explainer. Welcome back to the Explainer. Today, we're diving straight into the incredibly complex, but honestly, entirely logical embryology of the urinary system. For second-year medical students, getting a grip on these developmental stages is absolutely crucial for making sense of the clinical presentations you're going to see on the wards. Let's decode the origins of the human body together. Here's our game plan for today. We're going to cover the ascent of the kidneys, check out some renal, positional, and vascular anomalies, look at the division of the cloaca, trace the development of both the bladder and the urethra, and finally, wrap up with urachal and bladder defects. Let's get to it. Section 1. Ascent of the kidneys, from the pelvis to the abdomen. You know, it's pretty wild to think that our kidneys don't actually start out in the upper abdomen. Their initial home is way down in the pelvic region. So to get up to the abdomen, they have to pull off 3 super precise spatial movements. First, there's a cranial shift, moving from the 4th lumbar vertebra all the way up to the level of L1 or T12. Second, they do a lateral shift, moving outward to meet the adrenal glands. And third, the grand finale is a 90-degree rotation. The renal pelvis starts off facing forward, but rotates to face the midline of the body. So, what's actually driving this upward trek? Well, it's mostly caused by the diminution, or essentially the straightening out, of the embryo's body curvature, right alongside some really rapid body growth in the lumbar and sacral regions. The lower body is literally just growing away from the kidneys. But here is the really fascinating part, the vascular shift. The kidney is basically climbing an arterial ladder. Down in the pelvis, it's being fed by early pelvic branches of the aorta. As it climbs, it constantly grabs onto new, higher arterial branches, while those older, lower vessels just degenerate and disappear. Moving on to Section 2, renal, positional, and vascular anomalies. What happens when normal ascent fails? Normal development is like a highly choreographed dance, and if a step gets missed, the results are pretty profound. The most direct failure here is what we call a pelvic kidney. If the metanephros, which is the precursor to the permanent kidney, completely fails to make that cranial shift we just talked about, it gets trapped. It just stays right in its original embryonic location, deep down in the pelvis, situated right next to the common iliac artery. Now, I want you to mentally highlight this number, 1 in 600. That is a surprisingly high frequency for a congenital anomaly. It basically guarantees you're going to encounter this either on the wards or on imaging during your career. And that brings us right to the horseshoe kidney. As the developing kidneys get squeezed close together while passing through the arterial fork, their lower poles can actually touch and fuse. So now you've got this U-shaped kidney trying to ascend, but there's a major roadblock. Those fused lower poles physically hook right under the root of the inferior mesenteric artery. It literally blocks the ascent, permanently stranding this horseshoe kidney at the level of the lower lumbar vertebrae. Beyond just positional errors, vascular anomalies are super common too. Remember that arterial ladder we mentioned? Well, we frequently see accessory renal arteries when those lower embryonic vessels, the lower rungs of the ladder, simply fail to degenerate. These extra arteries sprout right off the aorta and usually enter the superior or inferior poles of the kidneys. So if you're ever reviewing an angiogram or scrubbing into a nephrectomy, you always have to hunt for these extra vessels. All right, Section 3, Division of the Cloaca, happening between weeks 4 and 7. Let's shift our focus back down into the pelvis where a massive remodeling job is taking place. Think about the central descending tissue structure, the urorectal septum. It physically grows downward to divide the cloaca. It essentially chops that single chamber into two totally separate compartments. Up front, you get the primitive urogenital sinus, which is going to handle the urinary tract. In the back, you get the hindgut or anorectal canal, which takes care of the GI tract. Now, here is a high-yield detail you absolutely must remember. This urorectal septum is formed by a layer of mesoderm. And what's more, as it finishes its downward division, the very tip of this septum anchors itself and ultimately forms the adult perineal body. That's a massive connective tissue hub in the pelvic floor, so definitely lock that fact in. Section 4, Development of the bladder, specifically the first part of the urogenital sinus. So we've got our anterior compartment set up. Let's trace the fate of this primitive urogenital sinus. We usually divide it into 3 distinct parts. That first, uppermost, and physically largest part, it is destined to expand rapidly. As it dilates and grows, this exact tissue forms the entirety of the urinary bladder. Initially, this developing bladder is continuous with an embryonic tube called the allantois. But as development chugs along, the allantois obliterates. It basically collapses down into this thick, fibrous cord that we call the urachus. The urachus acts as a tether, connecting the apex of the bladder straight up to the umbilicus, your belly button. And when you hit the gross anatomy lab, you'll recognize this exact structure as the adult median umbilical ligament. Moving right along, Section 5, Development of the urethra, the phallic part of the urogenital sinus. Okay, so we knocked out the upper part of the urogenital sinus. We're going to skip over the brief middle pelvic part for now and follow the anatomical path downwards to the third and final section, the phallic part. This is what gives rise to the urethra. And here's a foundational gold star rule of embryology to keep in mind. While the inner lining of the urethra is endodermal, the surrounding connective tissue and the smooth muscle that wrap around and control it, that is derived entirely from the visceral mesoderm. Up until now, development has been pretty uniform across the board. But by the end of the 3rd month, things diverge dramatically based on biological sex. In males, the epithelium lining the newly formed prostatic urethra starts to aggressively proliferate out into the surrounding mesenchyme. This forms the prostate gland. In females, we see a very similar budding process from the cranial part of the urethra, which ends up giving rise to the urethral and periurethral glands. Which brings us to Section 6, Urinary and bladder defects and their severe clinical correlates. When the beautifully orchestrated steps we just covered fail, we start seeing some pretty significant clinical malformations. Think back to the allantois we talked about earlier. Remember how it's supposed to completely obliterate into a solid cord? Well, if it doesn't, we've got a problem. If just a tiny, localized area of the allantois persists, the secretory lining inside will keep producing fluid. This creates a fluid-filled pocket called a urachal cyst. On the flip side, if the lumen in the upper part, right near the belly button, stays open to the outside world, it forms a draining sinus tract, a urachal sinus. Finally, let's talk about an exceptionally rare number, 1 in 30,000. This represents the incidence rate for one of the most severe multi-system failures in early embryogenesis, and it actually ties together a lot of the concepts we've explored today. We're talking about a severe ventral body wall defect. It's a massive failure of the lateral body wall folds to progress and close properly. We're talking way more severe than just an isolated bladder extrophy. Because the fundamental architecture of the pelvis is left completely open, that urorectal septum we studied earlier, it simply can't develop right. That leads to severe anal canal malformations, like an imperforate anus. And it doesn't stop there. Because the lower body folds remain unfused, the genital swellings get pulled widely apart, causing complex defects of the external genitalia. It literally merges urinary, gastrointestinal, and genital anomalies into one incredibly complex clinical presentation. So how do these microscopic, week-by-week embryonic migrations ultimately dictate a lifetime of human physiology and clinical outcomes? It leaves us with a pretty fascinating final thought, doesn't it? The difference between perfect health and a lifetime of complex pathology often comes down to just a few microscopic millimeters of mesoderm migrating exactly on time in a span of just a few weeks in utero. It's absolutely wild. Thanks for joining me on this explainer, and I'll catch you in the next one. Welcome to the Explainer. So glad you're here. Look, if you're a second-year med student, you already know embryology can kind of feel like this giant moving puzzle. Well, today, we're going to put some major pieces together by tackling genital system embryology. Specifically, we're looking at the exact step-by-step timeline of gonadal development. We're going to trace the morphological changes, the cellular migrations, and those all-important genetic triggers that literally dictate how we develop. Let's get right into it.
Here's our roadmap. We're tracking development chronologically today. 1. The indifferent stage. 2. Primordial germ cell migration. 3. Genetic sex determination. 4. Primitive sex cords. 5. Testis cord development. And 6. Seminiferous tubule maturation.
Starting off with part 1, the indifferent stage, our morphological baseline.
Okay, so the absolute most crucial thing to grasp here is the timeline. You know genetic sex is locked in right at fertilization, right? It all comes down to whether that fertilizing sperm is packing an X or a Y chromosome. But here's the catch. Morphologically speaking, nothing actually changes until the 7th week. That means for those first 6 weeks of development, male and female embryos are physically indistinguishable. It's wild, right? The whole genital system, the gonads, the ducts, external genitalia, they all go through this indifferent stage where they've got the exact same potential to go male or female.
Now, let's break down where these indifferent gonads actually come from. This is super high yield for your boards, by the way. They pull from 3 primary sources. First up, the genital or gonadal ridges. These start out as a pair of longitudinal ridges on the medial side of the mesonephros, and they're going to give rise to the capsule, the stroma, and the connective tissue of the gonads. Source number 2 is the coalomic epithelium. This covers those ridges and forms the primitive sex cords. Later on, those become your Sertoli cells in males or follicular cells in females. And finally, the third source, the primordial germ cells. These guys start way out in the wall of the yolk sac and eventually become the actual gametes, your spermatogonia or oogonia. Which brings us perfectly to part 2, primordial germ cell migration. An epic journey. We really need to track the exact chronological progression of these primordial germ cells because precise timing is everything on exams. Week by week, it goes like this. In the 3rd week, they're just chilling way out in the wall of the yolk sac, right near the allantois. By the 4th week, they start to move. They use this amoeboid creeping movement to travel right along the dorsal mesentery of the hindgut. By the start of the 5th week, they finally hit the primitive gonads. And then, in the 6th week, they actually invade the genital ridges. Just remember, the genital ridges have absolutely 0 germ cells in them until that 6th week. Check out this pathway marked in red. You can actually trace the primordial germ cells starting way down in the yolk sac. From there, you see them using that amoeboid movement to creep right along the hindgut, climbing up the dorsal mesentery, and finally settling into the genital ridge, sitting right next to the mesonephros. It is a seriously coordinated cellular migration. So why does this big migration matter so much? Well, let me give you a massive clinical pearl right from the text. If they fail to reach the ridges, the gonads do not develop. No way around it. These primordial germ cells don't just passively arrive and set up shop. They actually exert an inductive influence on the surrounding tissue. They are the trigger that forces the definitive sex glands to form. No migration, no gonads. All right, part 3, genetic sex determination, the developmental fork in the road. So, the germ cells have arrived. Now what? This is where we diverge from that indifferent stage, and it all hinges on the Y chromosome. If those migrating germ cells are carrying an XY sex chromosome complex, boom, male development is triggered. The Y chromosome has this specific gene, the SRY gene, which cranks out a protein called the testis determining factor, or TDF. Male development absolutely requires this active influence of TDF.
On the flip side, if there's no Y chromosome, there's no TDF. And without it, the embryo just defaults right into established female development. Oh, and quick pro tip for your exams. You need to know the exact address of that testis-determining SRY gene. It's located on the short arm of the Y chromosome, formally designated as YP11. Definitely commit that one to memory.
Moving to part 4, primitive sex cords. Let's look at what's physically happening in the genital ridge right around the time those germ cells are showing up and genetic determination is kicking in. The surface epithelium of the ridge starts proliferating like crazy. These cells literally dive down, penetrating the underlying mesenchyme. You see those darker orange clusters? Those form these irregular shapes we call the primitive sex cords. Now keep in mind, right at this exact moment, whether it's an XX or XY embryo, these cords are still completely tethered to that surface epithelium. If you were looking at this under a microscope right now, you literally couldn't tell a male from a female gonad. They look exactly the same.
Let's jump into part 5, testis cord development. We're following the male pathway now.
All right, so we're under the influence of the SRY gene and the testis-determining factor. Here's the step-by-step cascade. First, those primitive sex cords keep proliferating, pushing way deep into the medulla of the gonad. Second, they mature into definitive testis cords. You'll also hear them called medullary cords. Third, as they push toward the hilum of the gland, they start to break up into this network of tiny cell strands. That network will eventually become the tubules of the retate testis. And fourth, a thick layer of connective tissue develops, the tunica albuginea. This is huge because it permanently separates those deep testis cords from the surface epithelium. Take a look at that dense white band just beneath the surface. That is your newly formed tunica albuginea, and it is a real game changer structurally. Because of this dense layer, those horseshoe-shaped testis cords are now totally isolated from the outer surface. You can also see how the testis cords are reaching down to make contact with retate testis cords near the hilum. Let's zoom in and unpack this a bit more. Inside those horseshoe-shaped seminiferous cords, you've got two main players. You have the prospermatogonia. Those are from the migrating germ cells we tracked earlier, and you have the Sertoli cells, which, remember, came from the surface epithelium. Now look outside the cords. Floating out there in the mesenchymal matrix, you'll spot the Leydig cells. These guys are derived straight from the original mesenchyme of the gonadal ridge.
And finally, part 6, seminiferous tubule maturation. From embryo all the way to puberty.
Before we look at the final timeline, we really need to lock in how vastly different these structural pathways are. It's essentially a mirror image. In an XY embryo, the medullary cords develop into testis cords. Cortical cords are no-show, and you get that massive, thick tunica albuginea. But in an XX embryo, total opposite. The medullary cords completely degenerate, cortical cords spring up on the surface to hold the eugonia, and there is zero tunica albuginea.
You can really see this profound structural divergence by week 8. Look at the male side, deep medullary cords and that thick tunica albuginea creating a solid barrier. Then look at the female side. The medullary tissue in the center is degenerating, and instead you've got proliferating cortical cords right up on the surface epithelium, and absolutely no tunica albuginea separating them. Like I said, it's night and day.
Okay, let's wrap this up with some highly tested chronological milestones for the male system. By the 8th week, those Leydig cells hanging out in the mesenchyme wake up and start pumping out testosterone. By the 4th month, the testis cords take on their classic horseshoe shape, and the ends become continuous with the retate testis. Now, pay attention to this next part. It's super important. These testis cords remain 100% solid throughout all of childhood, literally completely solid. It isn't until puberty hits that these solid cords finally acquire a lumen. Once they hollow out, they get an upgraded name. They are officially canalized seminiferous tubules.
Which brings us to a pretty fascinating puzzle to leave you with. Once these seminiferous tubules finally canalize at puberty and are actually ready to do their job, how do they physically connect to the rest of the genital duct system? I mean, how do they link up with those mesonephric and paramesonephric ducts to actually transport sperm out of the testis? The embryonic plumbing has to connect absolutely perfectly for this to work. It's an incredible process, and we are going to explore the embryology of that excurrent duct system in our next explainer. Thanks so much for hanging out and learning with me today. Keep crushing it in your studies, and I'll catch you next time.
Welcome back, second-year med students. Today we're jumping right into the second half of our embryology explainer, focusing heavily on ovarian and genital duct development. Now I know, Embryology can sometimes feel like this crazy maze of, disappearing cords and transforming ducts. But stick with me here, because by the end of our time today, you're going to have a crystal clear, highly visual understanding of how biological sex diverges in the developing embryo. And more importantly, we are going to lock down those high-yield board exam points that examiners absolutely love to go after. Let's get to it.
Okay, here's our roadmap for this explainer. First up, development of the ovary. Then the different genital duct stage, followed by the hormonal regulation of those ducts. After that, we'll map out the male genital duct derivatives, and we'll wrap the whole thing up with a rapid-fire high-yield exam revision.
Section 1. Development of the ovary, looking specifically at female gonad formation. Female gonadal development is a really fascinating story of transition. And to totally cross this, you've got to anchor yourself to a specific timeline. So, initially, primitive sex cords basically break apart into these cell clusters right in the medullary part of the ovary. But, and pay close attention here, those early medullary cords, they eventually disappear completely, and they get replaced by a vascular stroma. And that stroma actually becomes the true ovarian medulla. So the real action for the definitive ovary actually kicks off in the 7th week. That's when the surface epithelium gives rise to a second generation of cords, called the cortical cords. These guys penetrate the underlying mesenchyme but kind of hang out close to the surface. Fast forward to the 3rd month, and those cortical cords split into isolated cell clusters that surround the primitive germ cells, the oogonia. Let's visualize that. If you look right into the center of the developing gonad here, you can literally trace those degenerating medullary cords making way for that vascular stroma we just talked about. Now cast your eyes outward, toward the edge, the surface epithelium. See how those cortical cords have proliferated near the surface and broken up into those distinct clusters containing the primary oocytes? This spatial relationship, where the medulla is degenerating on the inside while the cortex is just flourishing on the outside, is an absolutely foundational concept in embryology.
And that brings us to a standard classic textbook definition that you absolutely must have memorized for your boards. By the 3rd month, as those isolated cell clusters keep proliferating, they start to surround each oogonium with a single layer of epithelial cells. We call these follicular cells. Together, the oogonium plus its surrounding follicular cells constitute a primary follicle. This right here is the structural, functional unit of the developing ovarian cortex.
Moving on to Section 2, the indifferent genital duct stage, where we look at genital duct origins.
So before any hormonal divergence even happens, it's really cool to realize that every embryo essentially starts with a blank slate. Both male and female embryos possess 2 distinct pairs of genital ducts simultaneously. You've got the mesonephric ducts, historically called the Wolffian ducts, and the paramesonephric ducts, also known as the Mullerian ducts. Both of these pairs sit neatly right on the anterolateral surface of the urogenital ridge, and the paramesonephric duct actually arises right there as a longitudinal invagination of the epithelium. Let's spatially trace the path of that paramesonephric duct, because the anatomy here is super important. Start at the very top, the cranial end. Notice how the duct opens directly into the abdominal cavity with this really distinct, almost funnel-like structure. Now follow its path downward. Caudally, it first runs lateral to the mesonephric duct. Then, and this is a crucial anatomical move, it crosses the mesonephric duct ventrally and caudomedially. Finally, right in the midline, it meets its partner paramesonephric duct from the opposite side. Their combined caudal tip actually projects into the posterior wall of the urogenital sinus, and that creates a tiny little swelling known as the paramesonephric or Mullerian tubercle. Section 3. Hormonal regulation of the ducts, which drives the actual divergence. Now, what's really interesting about this step is how beautifully contrasting the hormonal pathways are. Let's say the embryo is genetically male. The testes take total control. The Sertoli cells start producing Mullerian-inhibiting substance, or MIS. And, exactly as the name implies, MIS actively suppresses those paramesonephric or Mullerian ducts, causing them to just degenerate. Meanwhile, the Leydig cells are busy pumping out testosterone. This testosterone directly stimulates the mesonephric, Wolffian ducts to develop into the full male reproductive tract. So one suppresses, the other stimulates. It is an incredibly precise biological handshake.
Section 4. Male genital duct derivatives, specifically looking at mesonephric origins.
We really need to draw a sharp contrast here, guys, because this is a classic exam trap, distinguishing between the epigenital and paragenital tubules. As the mesonephros regresses, a few of the excretory tubules, the epigenital tubules, manage to successfully establish contact with the cords of the rete testis. These are going to become the efferent ductules. However, the excretory tubules along the caudal pole of the testis are the paragenital tubules.
They do not join the retate testis. Instead, their vestiges just sort of hang out and remain there, collectively known as the paradiddinus. Please keep these two distinct in your mind. Epi connects, para misses.
Let's trace this spatial journey from top to bottom just so you can mentally picture the continuous transformation of the mesonephric duct. First, as we just established, the epigenital tubules form the efferent ductules. Second, immediately below where those efferent ductules enter, the mesonephric ducts undergo this massive elongation. They get super convoluted and form the ductus epididymis. Third, from the tail of the epididymis all the way down to the outbutting of the seminal vesicle, these ducts obtain a really thick muscular coat. This highly muscularized tube is the ductus deferens. Finally, the region of the duct that continues just beyond the seminal vesicles, that becomes the ejaculatory duct.
And this brilliantly illustrates that whole flow. The male genital duct system runs seamlessly from the retate testis out through the efferent ductules, winding all the way through that highly convoluted epididymis, and then traveling straight up the thick muscular vas deferens. But look closely at the cranial end of the mesonephric duct. See that tiny vestigial structure left over? That's the appendix epididymis. It represents the most cranial portion of the mesonephric duct that didn't form part of the main genital tract. So what happens to the paramesonephric ducts in the male? Well, because of that Mullerian inhibiting substance we talked about earlier, they degenerate almost entirely. But there is one absolutely critical exception you have to know. A tiny, tiny portion at their extreme cranial end actually persists. This remnant is known as the appendix testis. You've got to be able to differentiate this clinically and anatomically from the appendix epididymis we just looked at. Just remember, the appendix testis is a Mullerian remnant, while the appendix epididymis is Wolffian.
Section 5. Let's do a quick high-yield exam revision to lock this all in. Okay, rapid fire time. Here are your 5 absolute must-know facts from today's explainer. Number 1, by the 3rd month, the primary follicle is fully constituted. That is an ogonium plus its surrounding epithelial follicular cells. Number 2, spatially, always remember the paramesonephric duct crosses the mesonephric duct ventrally and cotomedially. Number 3, Sertoli cells secrete MIS to suppress the Mullerian ducts, while Leydig cells use testosterone to stimulate the Wolffian ducts. Number 4, the paragenital tubules at the caudal pole fail to connect to the retate testes and become the vestigial paradidymis. And number 5, the lone male remnant of the paramesonephric duct is the appendix testis. If you memorize these 5 points, you will absolutely secure those board points. To wrap things up, I want to leave you with a final thought. Something to sort of bridge your fundamental embryology with the clinical pathology you're going to see later. We've seen how perfectly orchestrated this whole divergence is, right? But if normal ductal divergence relies on such unbelievably precise hormonal triggers, what anatomical anomalies arise when MIS is totally absent? Or what happens when testosterone receptors are completely insensitive during that crucial indifferent stage? Seriously, consider how a genetic male might actually present phenotypically if these pathways fail to communicate. It's a fascinating puzzle. Keep that provocative question in mind as you move forward in your studies. Thanks for joining me for this explainer and happy studying. All right, future doctors, welcome to another explainer. I am incredibly excited to guide you through today's topic. We are taking a highly focused, high-yield journey through the embryology of the genital system. We're going to look specifically at how these vital structures originate, and equally importantly, the clinical outcomes when these developmental pathways go off track. This is pure, testable board exam gold, so let's get right into it. Here's our roadmap for this explainer. We'll start with female genital ducts development, move into the dual origins of the vagina, and look at uterine and vaginal defects. Then we'll switch gears to the descent of the testes, touch on hernias and cryptorchidism, and wrap up with the descent of the ovary. Let's get started. Okay, let's dive into Section 1, female genital ducts development. Take a look at the paramesonephric ducts. These are absolutely crucial because they're going to develop into the main genital ducts in females. Initially, you can spot 3 distinct parts to each duct. First, there's a cranial vertical portion, and that opens straight into the abdominal cavity. Below that, you've got a horizontal part that actually crosses right over the mesonephric duct. And finally, there's a caudal vertical part at the bottom. Now, as development continues and the ovary descends, something really important happens. Those first 2 parts we just talked about, the cranial vertical and the horizontal part, they become the uterine tube. Meanwhile, those lower caudal vertical parts actually fuse together right in the midline. This fusion forms the uterine canal and the paramesonephric tubercle. And here's a massive high-yield detail for you. As the female system develops, the mesonephric system regresses.
In fact, the only piece that sticks around is the epiophron, which you can see nestled right there in the mesovarium.
Moving on to Section 2, Dual Origins of the Vagina.
The way the vagina forms is honestly pretty wild. It starts as solid tissue that eventually hollows out. It all begins with two solid evaginations called the synovaginal bulbs, which grow out from the urogenital sinus. These bulbs proliferate and merge into a solid mass we call the vaginal plate. Then, a process called vacuolization kicks in, literally hollowing out this solid plate. By the 5th month of development, which is a major milestone you should definitely know, this outgrowth is completely finished and fully canalized into one continuous lumen.
And this exact process is why we say the vagina has a dual origin. Board examiners absolutely love testing this. Because of how everything meets and canalizes, the upper portion of vagina, including the fornices, comes from the vacuolization of the uterine canal. That means it has a mesodermal origin. But the lower portion, that comes from the vacuolization of the sinovaginal bulbs, making it endodermal in origin, specifically from the urogenital sinus. So you've got two totally different embryological origins fusing seamlessly into one single functional tube.
Which brings us to Section 3, Uterine and Vaginal Defects. So, what happens when that seamless fusion doesn't go exactly to plan? Well, a lack of fusion in the paramesonephric ducts leads to specific duplication anomalies. This can happen just in a small localized area, or along the entire line of fusion. Let's walk through the variations. First, you have the extreme case where there's a complete lack of fusion. This results in an entirely double uterus, called uterus didelphys, and even a double vagina. On the milder end of the spectrum, the uterus might just be slightly indented right in the middle. We call that uterus arcuatus. And then there's a relatively common anomaly called uterus bicornis, where the uterus literally has two distinct horns that empty into a single common vagina.
All right, let's switch gears completely for Section 4, Descent of the Testes.
As you know, the testes develop way up in the abdomen and have to make a massive migration downwards. This whole journey is precisely controlled by three main factors. Number one, the outgrowth of the extra-abdominal portion of the gubernaculum. Now, the gubernaculum is a mesenchymal condensation that extends from the caudal pole of the testis down into the inguinal region. When it grows, it triggers that initial intra-abdominal migration. Number two, as organs grow, intra-abdominal pressure increases, literally pushing the testis through the inguinal canal. And number three, the extra-abdominal portion of the gubernaculum actually regresses, pulling the testis all the way down into the scrotum.
You've got to know the timeline for this descent inside and out for your boards. Normally, the testes reach the inguinal region by about 12 weeks of gestation. Then, they migrate through the inguinal canal by 28 weeks, and finally, they arrive at their ultimate destination in the scrotum by 33 weeks. 12, 28, and 33 weeks. Definitely commit those milestones to memory.
Now, what's really interesting here is how the surrounding tissues react to this descent. Take a close look at the peritoneum. It actually forms a pocket called the processus vaginalis. This pocket follows the exact same path as the gubernaculum down into the scrotal swellings. And here's an amazing fact. As the testis drops down through the inguinal ring to end up in the scrotum at birth, it keeps its original vascular supply. Because it started high up in the lumbar region near the aorta, those testicular vessels have to stretch all the way down the abdomen alongside the descending testis. As the testes pushes its way out of the abdomen, it wraps itself in layers derived directly from the anterior abdominal wall. It's kind of like putting on coats as it leaves the house. Let's peel these layers back methodically. The innermost layer, the transversalis fascia, gets pulled down to become the internal spermatic fascia. The middle layer, which is the internal abdominal oblique muscle, gives rise to the cremasteric fascia and muscle, and finally, the outermost layer, the external abdominal oblique muscle, forms the external spermatic fascia.
So at this point, the testis is fully ensheathed in the scrotum. Remember that processus vaginalis we just talked about? It ends up forming the visceral and parietal layers of the tunica vaginalis, wrapping the testis in a nice protective serous sac. Now, this is crucial. The narrow canal that connected this vaginal process all the way back up to the peritoneal cavity normally obliterates right around birth or very shortly after. It's supposed to completely seal the system off.
But what if it doesn't seal off? That brings us to Section 5, hernias and cryptorchidism. Remember that connection between the abdominal cavity and the processus vaginalis that's supposed to close up? If that passageway stays entirely open, an intestinal loop can actually slip straight down into the scrotum. This is the exact anatomical basis for a congenital indirect inguinal hernia. On the flip side, sometimes the obliteration of this passageway is just irregular. It might close in some spots but leave small, open cysts along the track. Later on, these cysts can secrete fluid and expand into what we call a hydrocele. Another major clinical scenario is when the descent mechanism itself fails. In about 97% of male newborns, the testes are already in the scrotum before birth. For most of the remaining 3%, descent finishes up during the first 3 months after they're born. But in less than 1% of infants, one or both testes fail to descend entirely. We call this cryptorchidism. It's often caused by decreased production of androgens, specifically testosterone. The huge clinical takeaway here? Undescended testes failed to produce mature spermatozoa because the internal body temperature up there is simply too high for them. All right, let's bring this all full circle in our final section, descent of the ovary. Just like the testes, the ovary originally develops way up high in the abdomen and has to undergo an internal descent to reach the pelvis. But notice the structural difference here. The ovary's descent is actually arrested. It just stops in the pelvis. Why does that happen? It's because the female gubernaculum attaches directly into the side of the developing uterus. So instead of pulling the ovary entirely out of the body, it effectively anchors it right there in the pelvic cavity. Because of that midpoint attachment, the female gubernaculum ends up forming 2 distinct anatomical structures. The part that connects the ovary to the uterus becomes the ligament of the ovary. The rest of it, the portion extending from the uterus, running through the inguinal canal, and finally terminating down in the labia majora, becomes the round ligament of the uterus. It is literally the exact same embryological tissue as in the male, just anchored in a completely different way. So, as we wrap up, think about this. We've just seen how the exact same starting materials, things like the gubernaculum or the mesonephric ducts, are masterfully repurposed by the embryo to build entirely different, highly specialized tracts. It really begs the question, how does a single layer of mesenchymal tissue dictate the fate of entirely different organ systems across biological sexes? It's honestly a testament to the mind-blowing precision of human development. Review these pathways, understand those clinical correlates, and you are going to be perfectly primed for your board exams. Thanks for joining me on this explainer, and keep studying hard.